Thank you, Bill. I'm very pleased to introduce our next speaker, Dr. Warman, from Boston. And he is a geneticist and also works in orthopedic surgery. You know, when I started in this area several years ago, perhaps because my wife is also a geneticist, I was very interested in figuring out the genetic basis of these vascular malformations. And many people are confused between what's hereditary and what's genetic. And not everything that's genetic is hereditary. And almost all human disease, we're going to find out, is genetic. There's the abnormal genes that cause disease. What's been very exciting over the past 25 years is that people, particularly our colleagues in Boston, have been starting to identify specific genetic mutations for a variety of vascular malformations. And we'll see if Dr. Warman agrees with this, but I suspect that in the next 20 years, we'll have completely different names for all these vascular malformations. Instead of separating them by what they look like, we're probably going to separate them by what genetic mutation is responsible for it. But we'll let an expert address that. Now, I need to figure out, see this came off.
Yeah,
You know what to do?
I think I do. So let's see if this will work. You need a young person to really make this work. There we are, right there. Thank you. How much time do I have? Because we're running a little bit behind schedule, so I don't want to overstay my welcome. And also, would people like to stretch? Because you've been sitting on your bottoms for about an hour and a half now, I think. So
We usually do run overs. We usually do run over, so we'd rather hear what you have to say.
okay. So first of all, thank you for inviting me out to Rochester, Minnesota, where I've never been to before. And I hope to visit with all of you and to see a little bit of the city before I return back to Boston. What I want to do in the next 30 minutes or so is tell you about the the progress we've made in understanding the genetics of Klippel Trenaunay syndrome, with the goal of putting the surgeons and the dermatologists out of business. Meaning that if we can identify the genetic cause, and from the understanding of the genetic cause, we can understand the physiology or the biology that underlies the malformation, the overgrowth, the fatty changes that can occur. Perhaps we could use more targeted therapies, pharmacologic therapies, rather than surgical therapies, to try to treat as much of the symptoms and signs of the disease as we can. Now, this might be 10, 20 years off, but this is the ultimate goal. And the surgeons will tell you that they would be more than happy to be put out of business doing this, because there's plenty of other things for them to do. So I don't think we're going to get much slack. I want to just begin by thanking the folks in my lab whose work I'm going to tell you a little bit about today. And that would be Rudy Murillo, Joanna Lopes, Ora Reichert, Stephen Hahn, just to name a few. Okay, so what did Dr. Driscoll mean by genetic diseases? So genetic diseases, I talk about it, is a change in the DNA. So if we look at a typical cell, which might be here, and here's the nucleus, and within the nucleus are the chromosomes, and the chromosomes are made up of double-stranded DNA. By genetic, I mean a mutation that affects the DNA itself. So genetic diseases are due to alterations in DNA. Now, when we think about genetic diseases, most of us think about hereditary genetic diseases. I've not watched the show, but some of you may know the show from the Learning Channel. I think this is Little People, Big World. And I cut off the dad, but this is the mom, and she has achondroplasia. And this is one of her children who has achondroplasia. So this is mom, the circle, dad, the square. This is her son here who has achondroplasia. And then she has 2 children that do not have achondroplasia. So one mutation in the gene FGFR3 is sufficient to cause achondroplasia. And if you pass on that altered gene, your child will inherit achondroplasia. Now here's another example of a genetic disease. This is albinism. So this African-American couple each carry a non-working gene that affects skin pigment formation. And while they have one working copy of the gene, so their skin is normally pigmented for an African-American, this child right here, shown here, got both copies of mom and dad's non-working genes. And so she is unable to make skin pigment, and so she has albinism. So again, mom and dad have non-working genes, they have mutations in their DNA, and they pass on the mutation in double dose to this daughter, whereas these children did not get the double dose of the mutation, and therefore they're not albino. So what both of these families have in common is that they have a mutation that was passed through egg or sperm. In this family's case, mom, in her egg, gave a mutation that went to this son. And in this family's case, dad and mom, in sperm and egg, passed on a mutation. And we call these mutations germline mutations. They go through the germ cells. The germ cells meaning the cells that form sperm and egg. Now, there are other genetic diseases, and this is an extreme example, that is not hereditary, but is still caused by a mutation in DNA. And this is a poster of a woman who was a long-term smoker, and as a consequence of smoking's carcinogens, the carcinogens created a DNA change in one of her throat cells, and that cell became malignant, became cancerous, and caused it to overgrow, and as a result, she had to have her voice box surgically removed, and she has a hole there in which she's now breathing into her trachea. So she has a mutation in DNA that's caused her cancer, but she, shown here, did not inherit that mutation from her mom or dad, cannot have given it to any of her children, because the cells that were mutated were just in her throat. And they arose as a result of one cell becoming mutated due to the carcinogens. So this is what we call a somatic mutation. It's in the soma. It arises not in sperm or egg, but in the soma of the body, all the other cells of the body. And we know some things that can cause high rates of mutation, such as smoking, such as too many X-ray exposures, other carcinogens that are in the environment. But let's face it, we're all human. As humans, we are imperfect, and if we do not die of heart disease, or stroke, or, well, those are the 2 biggies. If we live long enough, one of our cells is going to become mutated and cancerous, and we're going to die of cancer. So I would say any human that is capable of living long enough is going to have one cell as an accident of nature become cancerous and kill that person. So it's not that all mutations are due to carcinogens in the environment, like smoking or other exposures. Most mutations just occur by chance. And every one of us in the room, we have trillions of cells, we have thousands of mutant cells in our body. Probably millions of mutant cells in our body right now. So it's not just having a mutant cell, but it's when that cell first became mutated and what that mutation was capable of making that cell do that determines what happens to that cell and its progeny. So the most common cause of non-hereditary diseases are these somatic mutations. And KTS is the result of a somatic mutation. Meaning that if you look at a person with KTS, a fraction of the cells in their body have a mutation that arose as an accident of nature. It did not come from mom or dad. It was not inherited through the germline. And it will not be passed on to son or daughter. It is a somatic mutation unique to that individual that arose as an accident of nature, as all humans are prone to having accidents of nature, and it is just within them. So here's the idea, that anyone with KTS, when they were an embryo and were 50, 100, 1,000 cells old, one of those cells developed a mutation as an accident of nature, and that cell's progeny, its daughter cells, I'm being sexist here. I guess not. Yeah, it is, daughter cells. I could say son cells, right? So its progeny, wherever those cells end up, are capable of causing a problem. So a mutation that arises de novo, out of nowhere, in an embryo, is a somatic mutation. It's not inherited in the germline from mom and dad. Now, Rudolf Hapley, he's a dermatologist. He said these sorts of somatic mutations, if they're significant enough mutations where they can't survive at every cell of your body because they would kill you, they're going to follow certain rules, and these are Hapley's sort of 5 rules. One is that the birth defects that they cause always arise sporadically, meaning they never come from mom or dad. Second, they're distributed in a mosaic pattern because that first cell, wherever its progeny cells end up, determines what the malformation or the problems are going to be. Third, the involvement is variable because a person whose first cell becomes mutated when they're 50 cells old will have 1 in 50 of all their cells in their body ultimately with a mutation, whereas a person that has the first mutation when they're 1,000 cells old will only have 1 in 1,000 cells with a mutation. So depending upon when that mutation arose, you'll either be more severely affected or potentially less severely affected. That you can never have your whole body affected, because if every cell in your body were affected, you could not survive. Just as people with cancer, not every cell in their body is cancerous. And in fact, when cancer cells have become so extensive, they can kill. So in the case of these types of somatic mutations, they're spotty and not in a whole liver or a whole leg or a whole body. And lastly, if you're an embryo and a cell becomes mutated, it shouldn't matter whether you're a boy embryo or a girl embryo. So males and females, boys and girls, are affected at the same rate. So this isn't just a girl's disease, this isn't just a boy's disease. KTS can affect men and women equally. So here's sort of what I mean by KTS follows Hapley's predictions. No patients with KTS have inherited KTS from a parent or passed it on to their children. They always occur sporadically. The involvement can appear mosaic. So here's an example of sort of a mosaic appearance of the vascular stain, but not all the tissue of the leg is vascular. Some individuals can have more severe involvement than others. Their mutation maybe arose earlier in embryonic life than other people's mutations. Males and females are affected equally. Now the idea that there can be a mutation that can kill you if it was in every cell of your body, but can survive if it is surrounded by wild-type cells or healthy cells, was first proven before Hapley back in 1978. And for the young people in the audience, 1978 was when the first test tube baby was made by in vitro fertilization. So this is Louise Brown, the first test tube baby born in 1978. So one could, back in 1978, take female egg, male sperm, put them together in a test tube, get an embryo, and put the embryo back into a mother so that she could carry that embryo to term and give birth to a child. Well, this mouse, called the hairpin mouse, HP, and that's the little hairpin, if you see the very end there, this was a female mouse that could not have babies. This mutation, the hairpin mutation, was lethal, meaning if she gave it in her egg to her offspring, the offspring would die in the womb. So she could not pass on this mutation. So what Dorothea Bennett did was she took the early embryo from this hairpin mouse. This embryo is all mutant cells, and it's going to die. And she mixed it together with a wild-type mouse embryo. So she made a mosaic, a mixture of mutant cells and wild-type cells. And this embryo, when put back into a pregnant mom, could survive, be born, and in fact could have the hairpin mutation, the same as mom. So this was proof that cells that would kill you if they were in 100% of your body can survive if they're surrounded, perhaps nourished, helped, by healthy cells. So again, this is proof that somatic mutations like this can exist in mice and in humans. So when we set out to identify the cause of Klippel Trenaunay syndrome, which I'm going to tell you we have found today, we did it with 3 hypotheses. Sorry, I have to see if I can make that thing go away. Anyone have any idea? Right click, there you go. Okay, now I got to go back to hypotheses. Okay, so the first hypothesis is that KTS malformations are the result of somatic mutation. This that I showed you already. And if we were going to look in the affected tissue, we would find some mutant cells. The second hypothesis is that if we wanted to find cells that have mutations, we want to look here rather than here, meaning affected tissue is going to be enriched, have many more mutant cells than perhaps unaffected tissue is going to have. And the third thing is, if we look really, really hard using technologies that were available, we started in 2010, I think, so 4 years ago. If we looked really hard, we should be able to find a mutation. And the way that we decided to look was we took affected tissue that either Dr. Fishman and his colleagues excised from patients, or Dr. Keurig and his colleagues in pathology had sitting in a tissue block from a biopsy that might have been done years before. We extracted DNA from the affected tissue. We sequenced the heck out of it, looking for mutations. And we compared the DNA in the affected tissue with DNA from what we hoped would be unaffected tissue, like blood or skin, that did not have a malformation. And we asked, what's in this tissue that's not in this tissue? So that was the idea. Look for mutations in affected tissue that are not in healthy tissue or unaffected tissue. And this is how we did it. And I have to stop here, and Dr. Fishman always likes when I do this, so you have to pay attention. This is my cell phone. It's incredible, right? I can cruise the internet, I can get phone calls, I can send text messages. I do not have a clue how this thing works. Most of you don't. I can use it, right? And I know some of the ways in which it makes mistakes. For example, I've been butt dialed, and people have butt dialed me. I've typed in something, and it's auto-corrected what I typed in to a word that you're laughing, but you know this, to something different than what I had intended, and now I sound like a jerk, right? I misdialed someone at 2 o'clock in the morning, or I called someone on the West Coast thinking that it was the same time as where I was on the East Coast. So we know what can go wrong, but I have not a clue how pushing the button sends a signal up to some satellite and reaches someone else. So even though I don't know how it works, I can use it and get something out. So I don't expect you to know precisely how sequencing works nowadays, but just know that we can do it, and I'm going to point out some of the difficulties in doing it as well. Okay, so this is how sequencing works in 3 easy steps. So the first thing you do is you just get DNA, and you can get DNA from affected tissue that's either been freshly obtained or in these paraffin blocks, and you can use a teeny tiny amount of tissue, because that contains thousands, millions of cells, which is more than you need. You then break that DNA into tiny little fragments that are 200 base pairs long. And what that means for you and I is for every cell, we're making, I think, 10 billion fragments of DNA. So take 10 billion fragments times 1,000 cells. That's an awful lot of tiny little bits of DNA. To that DNA, in a test tube, we add sequencing adapters, just a chemical reaction. And we can do this from fresh DNA, or we can do it from fixed DNA that's already from the fixation process been fragmented. So that's step 1. Get DNA, extract it, break it up into tiny little pieces, and add things for sequencing. Second, you send them to a sequencing facility. And again, if I go back to my cell phone, this cell phone takes pretty darn good pictures. Right? I could take a nice picture of the whole room here, so it has pretty decent resolution. So what a sequencing facility does is it takes those billions of fragments of DNA that have now been randomly broken up into tiny little pieces. It puts them on a glass microscope slide, separate it out. So here's one DNA fragment landed here, here's another DNA fragment landed here. And then it sequences each, and the fragments get stuck onto the glass slide. So it then does a little sequencing reaction. So it knows that this fragment is here on the glass slide, this fragment is here on the glass slide, and every time it asks the sequence of that DNA fragment, it takes a picture of it. Now it's taking millions of pictures because we have billions of fragments on this glass slide. So it's not my pretty good cell phone camera, it's a nice microscope camera that's just automatically going and doing like a million pictures in a pretty large field. But it works incredibly well. And with this technology, it's possible to sequence about 1 billion tiny little fragments at once at reasonable cost. So now you've sequenced on a glass slide 1 billion things, and you get your data sent back to you by the center. And you get your data sent back sort of like in an Excel spreadsheet, for those of you that know Excel. And Excel, it's got 2 columns in it. One column is the fragment number. So there's 250 million fragments, 1 to 250 million. And then each fragment, that's the DNA sequence of that fragment that the machine had figured out. And these tend to be about 100 base pairs long. So 250 million times 100 base pairs long, that's 25 billion pieces of DNA information, and our genomes are only 3 billion base pairs. So we can sequence ourselves about 8 times over. Pretty good for like $1,000 bucks. Of course, they're going to charge you more, but the material cost is like $1,000 bucks. Okay. So now, how do you image these Excel spreadsheets? And it's all done computationally, but I'm going to show you what the output sort of looks like if you wanted to visualize it. So this is a picture of a chromosome. I think this is, I don't remember what chromosome it is. It probably says up there, chromosome 12. And we're looking in, I think it's right here, this region, at about 100 base pair interval. That's shown right here. There's about 100 base pairs of DNA. And this is the reference sequence. This is some person considered normal. And this is what his or her DNA sequence looked like. And then the gray bar is one of those rows of that Excel spreadsheet. It's 100 base pairs of DNA, about. And if the DNA of that fragment is identical to the reference sequence, it's shaded gray. And if it differs from the reference sequence, as several of these fragments do here, they're color-coded. So the reference sequence says there should be a thymidine residue here, but there's a bunch of gray bars that say, oh no, there's a guanosine residue there. So this is finding someone that has like the AB blood type. The person who was sequenced down here might have just had the A blood type, and here you found, oh, but this person has the A blood type and the B blood type. So this is AB. And these changes here, this change here, is 1 bar with a mutation out of 29 bars that cover this. So this could be a somatic mutation, not inherited, that's present in 1 in 20 cells. So most cells have the normal sequence, which is why they're all shaded gray, but one cell gave a DNA fragment when it was made into those tiny little pieces that we sequenced that contained a mutation. So this is very hard to visualize by eye, because we have to do this for 3 billion base pairs of DNA. So we let computers do it. So it is the veritable looking for a needle in a haystack. And this was unimaginable 20 years ago. Even 10 years ago, there was not the technology. But there were clever people 10 years ago who made this a reality today. So using this needle in a haystack, let me show you the numbers of how we did it. So we took affected tissue and we sequenced, we focused on 50 million base pairs of all of our DNA that encodes genes and proteins. So we sequenced 38 million of those tiny little fragments from someone's affected tissue. And from their unaffected tissue, it was easier to get more DNA. We sequenced 86 million of their fragments. We got 100 base pairs for every fragment, so that's 3 billion base pairs of sequence, or every piece of DNA, we had 77 of those gray fragments overlapping. We did better for blood. We had 173 of those gray fragments overlapping. We asked, how many of those gray fragments contained one of those little color-coded changes that said, oh, this isn't the quote-unquote healthy person sequence, this is different from the healthy person sequence. We found a change every 41 base pairs. We found 1.2 million changes. That's a lot. You can't ask a student to go through them one by one and ask which is maybe the causal change. And the reason why we found so many changes is because this technology, like my cell phone, is not perfect. It does autocorrect, it butt dials on occasion, except the errors that this technology makes is it calls this as a mutation where it's not. It's a false positive. It's miscalling it. Because here's another example of a mutation that this is calling. So we have to find a way, we have to set some rules to not be distracted by what I would call these sort of one-off changes. So we can make some rules. You have to find the change twice. 2 gray bars have to have it as opposed to just 1 gray bar, for example. The change has to alter an amino acid or something that will change the function of the protein. And if we do that, we go from 1.2 down to 18,000. That's a reasonable number. Most of these differences still are going to be like the AB blood type. Brown hair, blonde hair, blue eyes, green eyes. So if we get rid of all the differences that people are walking around with already, that gets us down to only 690 DNA changes found in affected tissue from the person whom we sequenced. And this was 4 years ago. If we were to do this now, we'd probably only be down to about 69 changes, because we found more normal variation. And then you could say, okay, so how many of these differences were found in affected tissue and were not found in the blood? There was 31. And if we ask how many of these differences, these 31, were found in more than one patient with KTS? The answer was only 1, and that change was in a gene called PIK3CA. So by doing this sequencing, and this, I made it seem easy, but this was a lot of hard work. And it was built upon the hard work of other people who developed all these incredible technologies. But we found that PIK3CA mutations cause KTS, And the mutations cause this protein to function too well. So what does this protein do? So this protein is part of an enzyme called phosphatidylinositol 3 kinase. PI3K, easier, right? It lives at the surface of a cell membrane. The cell membrane is shown here. And when a growth factor binds to a growth factor receptor at the surface of the cell membrane, this enzyme turns on a series of cascades that ends up causing cell growth, cell proliferation, increased survival, and decreased death. So if you think about the features of Klippel Trenaunay syndrome and related overgrowth syndromes, increased cell growth, well, there are vascular malformations, there's lipomatous overgrowth, there's limb length discrepancy, there's your increased growth. There's also decreased cell death because the cells are dividing and they're not being matched with an equal rate of cell death in order to keep things from overgrowing. And the receptors that this enzyme is immediately downstream of are the vascular endothelial growth factor receptor, blood vessel formation receptor, the fibroblast growth factor, a skeletal growth factor, the insulin-like growth factor receptor. So a lot of the component features of Klippel Trenaunay syndrome are explained by cells that have this mutation, thinking that they are getting these growth factor signals when in fact they're not. So this mutation fools those cells into thinking that they've had a growth factor bind to the receptor, whereas instead this is just turned on. It's a leaky faucet, independent of whether there's any growth factor signaling. So if this cell was in an endothelial cell, it would think, oh, vascular endothelial growth factor is bound to its receptor, I need to make new blood vessels. If it's in a fat cell, oh, insulin-like growth factor is bound to the insulin-like growth factor receptor, I need to make more fat cells. Now, Dr. Adams is going to tell you about an inhibitor of mTOR signaling, and you'll notice that mTOR is downstream of this enzyme. So the efficacy of this inhibitor now makes some sense. Because if this enzyme is turning on this pathway and you block it over here, then that's one sort of pharmacologic way of helping prevent what this mutation is doing. And the way I look at this, for those old enough in the audience, or even the young ones, is the mousetrap game still out there? Yes, okay, so you know the mousetrap game where you put in the marble and it does all these things, or the old Rube Goldberg puzzles, right, the cartoons. This is sort of a pathway that gets activated by all those other things. Okay, so let me move ahead. Okay, so I told you about the hard work for finding mutations. It took us about a year to do. And I'd like to think we could do it faster now, but even a year in the grand scheme of things is not that long, given that this was unsolvable. So now, in the 4 years since we started, there's even newer technology that make these needle in a haystack searches easier. So in the old days, if you wanted to spread hay, you had to do it by hand. Now there's turbines that can spread hay or pick up hay, whatever you want to do. Nowadays, if you wanted to get a nice picture of your home from the air, this is maybe the resolution that you can get. Now you can figure out which trees are in bloom, whether you have cherry blossoms or apple blossoms or pear blossoms, right? So these are all improvements in technology. And these same improvements in technology that have occurred in the case of farming have occurred in the case of genetic studies. So here's one such improvement, where we can take 2,000 cells, put them into a little mixture in a test tube, and then break up this little mixture into 14,000 little droplets. So in a test tube now we have 14,000 droplets. This you should be thinking, oh, what's the big deal? He just showed me you can do 250 million fragments. What's 14,000 droplets? But the 14,000 droplets are cool because if you've only put in 2,000 cells, and each cell has 2 copies of DNA, then only 4,000 droplets will contain DNA from a gene of interest. And 10,000 droplets will have nothing in it. And then we can design a specific assay that could look at each cell, and if the cell contains a wild-type allele, that droplet will fluoresce red. And if it contains a mutant allele, that droplet will fluoresce green. And if it contains no DNA, there'll be no fluorescence, it'll be empty. So we can do this little reaction in the test tube. This is a $3 test. And at the end, here's the 14,000 droplets. Here's with wild type PI3 kinase genes. Here with mutant PI3 kinase genes. And then you just count the droplets. How many have nothing in them? How many have wild type allele? How many have mutant allele? And this is what it looks like in real life. So if you put in no DNA, these are all the droplets that have nothing in them. They're empty. If you put in wild-type DNA, these are all the droplets that have the wild-type allele. These are the droplets that have nothing in them. If you put in affected tissue from an individual with Klippel Trenaunay syndrome, these are the droplets, and I told you how many droplets are. About 8,900 have no DNA in them. About 3,800 just have the wild-type allele. 242 have the mutant allele. So that's a small fraction, that's less than 10%. And every now and then you get a droplet that gets one copy of the wild type, one copy of the mutant. So it sort of fluoresces somewhere in between. So using this technology, we now, for $15, can look for these needle in a haystack common mutations that occur in Klippel Trenaunay syndrome. And in 21 samples that we've looked at, we found mutations in 19. So that's about 90%. The same mutations that cause Klippel Trenaunay syndrome also cause a slightly more diffuse syndrome called CLOVES, congenital lipomatous overgrowth with vascular, epidermal, and skeletal anomalies. And they also cause a more localized malformation, an isolated lymphatic malformation, the old-fashioned cystic hygroma. So the same gene, the same mutation, can cause many different phenotypes, clinical situations. Why? When did the mutation arise? What cell did it arise in? And where did the progeny of those cells end up? Earlier mutations tend to be more cloves-like. Mid-embryonic development, whatever that means, may be more Klippel Trenaunay-like, and later mutations more isolated lymphatic malformation-like. Okay, so I told you that if you have typical, strictly defined Klippel Trenaunay syndrome, and by that I mean cutaneous stain, lymphatic malformation, persistence of embryonal vasculature, and with or without overgrowth, you are very, very, very likely to have a somatic, not inherited from mom or dad, not passable on to child, PI3 kinase mutation. Now, how are we going to study that in an animal model to put the surgeons out of business, to come up with drugs? So the way that we have to do it is we have to make a mouse that does not have the mutation in every cell of that mouse's body, because that will kill the mouse. So we have to make a mouse in which we can turn on the mutation in a fraction of cells, give this mouse a phenotype, and then throw drugs at it and ask which drugs work. And we can do that by quantifying the response. So this is our entree into doing it. So we have a mouse in which we can turn on the PI3 kinase mutation, and the distribution that we turn it on in is the skeletal elements of the arms and legs and the skin overlying the head and the appendages. So this is an unaffected mouse, and this is a mouse that has the somatic mosaic, the induced mutation, not in every cell of the body, but in the skin here, in the arms and in the legs. And you can see that we have blood vessel malformations, We have, it's hard to see, but there's an extra little pinky there. Some people with Klippel Trenaunay have an extra toe, sometimes an extra pinky. And we can create that in the mouse. And the limbs also are slightly overgrown. The problem we have with this mouse is that the moms eat them. Unlike human moms that are nice to their babies, mouse moms oftentimes eat all their babies anyway. So that it's been hard to study. But nevertheless, we can, even though the mice don't survive, we can see the blood vessel malformations underneath the skin of a mosaic mouse compared to a wild type mouse where the skin is more normally formed. And we can do the following experiment. We can create these embryos, and at embryonic day 9 or 10, we can give a drug to the mom. And I'm not advocating that pregnant women carrying children who have CLOVES or KTS or lymphatic malformation be given a drug, because these drugs may not be good for fetuses during development, but this is a proof of principle experiment. If it works in a mouse, then maybe we first try it in a child or in an adult, and then we work our way backwards in human patients. So we can get mice to have these somatic mosaic embryos. At a specific gestational age by ultrasound, we give them drug, and then we ask what happens. And we're testing FDA-approved drugs, including sirolimus and several other drugs that are in development. And we're also testing drugs that have already been through studies by drug companies, have failed for their current indication, and the companies are looking to repurpose them. They said, oh, we were hoping it would be useful for this, and it doesn't work. Would you try it in your animal model? But we're only using drugs that fall in the pathway. And this is sort of the granddaddy of the repurposed drug, Viagra. It was an antihypertensive medicine that did not work as a good antihypertensive medicine, but was great for erectile dysfunction, right? So it was repurposed. Second to last slide, third to last slide. We're confident that PI3 kinase mutations are the most common cause of Klippel Trenaunay, CLOVES, FAVA, which I haven't told you about, and isolated lymphatic malformations. So this child with a large lymphatic malformation of his neck, this is the same child after therapy, and same mutations as cause KTS. So in thinking about genetic testing for KTS, persons with KTS, you probably have a PI3 kinase mutation as a somatic change. We're not going to find it in your blood, unaffected tissue. There's 5 common mutations that we see that account for 80 to 90% of cases, and that's a $15 assay in our lab to do. We're not allowed to charge for it, so we only do it on a research basis. Mutant cells should be present in affected tissues, but if you send us unaffected tissues, we may not find it. We really need to look at affected tissues. There are labs, if you're dying to know and you want an official report back. There's CLIA, which is a pathology-approved fee-for-service company that offers testing for these common mutations. They also will require affected tissue. The problem is that more than 10% of the cells in that tissue that you send have to have the mutation. Otherwise, the commercially available tests are likely to miss them. So you don't want to spend a lot of money and get a negative result just because the sample that you sent wasn't the right one, the test wasn't sensitive enough. And that's particularly the case if you send, say, a blood vessel malformation or skin biopsy, because in our hands, fewer than 10% of cells are mutant in many patients. And then finally, I'm going to just do a little plug, because I brought the cups with me and the consent forms. And that is that there are tissues where we think these mutations can hang out and not cause huge problems, but one can get those tissues pretty easily. And one such tissue is the urinary system, the kidneys, the bladder, the collecting ducts. And so we are doing a project right now on a research basis, so it's give your P for science, to see whether we can find PI3 kinase mutations in the cells that you just normally pee out, that are in your renal system that just you pee out every day. And thus far, we've sequenced urine from 13 patients with Cloves syndrome, same mutation, a little bit more extensive involvement. And we've been able to detect mutations in 5 of their 13 urine samples. So if we find a mutation, it saves having to do a biopsy and a more expensive or invasive test. If we don't find a mutation, it doesn't mean you don't have one, it just means you're not peeing out. So with that, I just again want to thank the folks in my lab. I've got a fantastic lab. And Kyle, Val, Matt, and Nolan, who are not in this picture, were integral for doing this work. Patients and families who've contributed. The Vascular Anomaly Center at Boston Children's is one of the most fun experiences I've had, professionally and personally, being part of that. The Manton Center for Orphan Disease Research that's funded some of this work. Wayne Phillips has shared the mice. Steve McCarroll, this digital PCR technology. And I'm funded by the NIH and by the Howard Hughes Medical Institute. Thanks a lot for your attention.
Questions to Dr. Warman?
I have a question regarding Dr. Wayne's research associated with the ABCF1 gene coupled with vascular malformations that were very similar to Klippel Trenaunay and even actually associated with Klippel Trenaunay in certain family lines.
So remind me of those papers. Was that the paper that was published about 10 years ago in Science?
No, it was actually fairly recently. He even created a zebrafish model through overexpression of AGTF1. The zebrafish.
What's the name of the gene?
AGTF1, angiogenesis, some kind of angiogenesis factor. And so I'm kind of curious, could this, considering he did find there was a hereditary predisposition that was found to be statistically significant with that particular gene, could there be a double hit sort of going on, like in some families, like a predisposition manifested by a PI3 kinase mutation?
That's certainly a possibility. Again, I think critical is strictly defining what KTS is. So in his families, did they truly have KTS or do they have something different? Because we know that there are many other vascular malformation syndromes that are caused by genes other than PI3 kinase, RASA1 being one of them, say for Parkes-Weber syndrome, and TI2 for Blue-Weber-Blavivus syndrome. So I think that the strict definition of the syndrome is critically important. To get at your question whether genetic background might be a risk factor, because all of us are walking around with PI3 kinase mutations, I guarantee it, because we've got trillions of cells. Some of them are going to have these same PI3 kinase mutations, so why don't we have the same syndrome necessarily as our patients or family members who do have KTS? I think it's more likely that our mutations arose too far along in development to cause us huge problems. And had they occurred earlier in embryonic life, we might have had the same condition. But I can't exclude the possibility that maybe it's because our genetic background, all these other gene changes, are slightly different than from those individuals who have the clinical manifestations of KTS or Cloves or some of the other syndromes. But I'd be very interested in looking at that paper. I wasn't familiar with the paper to know. But of the KTS samples that we've looked at, strictly defined, greater than 90% have somatic PI3 kinase mutations. But that doesn't mean that this is by itself sufficient to cause the disease. Maybe genetic background is important, but in the mouse, turning on the mutation is sufficient to cause the vascular malformation and the overgrowth in the mouse.
Is there a way that you could actually look for both of those genes simultaneously, like one somatically and one,
So in fact, we could probably do that with the samples we have already. Because remember when we looked for the PI3 kinase mutation, the first needle in a haystack, we sequenced all the genes in the body. So we actually know all the sequence of those individuals, and we could go back and look specifically at this particular gene to see if these individuals have this gene altered as well, that gene altered as well. I think that would be a very good thing to do. So we should talk afterwards and take a look. Yes?
towards the beginning, you showed the lady with the laryngeal cancer problem, and the fact that was a somatic mutation, kind of brought on by presumably by the smoking. So with this, like I understand you're saying basically crap happens and this somatic mutation happened to cause KTS. But so is there any effort being made, or is it considered to be irrelevant what the smoking equivalent was for KTS?
Right, so I want to be very careful, clear here by saying that we know that certain things absolutely increase one's risk for getting cancer. There's certain chemicals, there's carcinogens in cigarette smoke. But even if you were to get rid of all of those things out of the environment, make our world carcinogen-free, we're still getting mutations. And the reason we're still getting mutations is because we've got 3 billion base pairs of DNA that we have to replicate every time our cell divides. And we're just not designed to do that very well.
And so we're just, science at this point is just prepared to say, we're going to live with the fact that those happen, and we're going to look at afterwards, are there drugs we can introduce to turn that off or whatever? We're not going to look at what caused that. In the way that we've said, you know, oh, we know that cigarettes cause cancer now.
Well, so people are interested in whether there are genetic backgrounds that make some people better at repairing their DNA than others, for example, as they're replicating it. To see if we can, we know that there's genetic mutations that make people worse at repairing their DNA. Many of the hereditary colon cancer mutations, for example. Cancers, you don't inherit the cancer, you inherit the repair defect that causes mutations to accumulate that then gives you cancer. But we know that there are mutations that cause DNA to repair less well. We don't know if there are some of us walking around with variations in our repair genes that are good, where we very rarely make errors in replicating our cells. I don't think that's going to be the case. Some might be a little bit better than others, but I think that difference is trivial compared to the trillions of cells we are over a lifetime. And the fact that, as you said, bad stuff can happen on occasion. So I think that people are looking at antecedents, but thinking about it in the case of KTS, remember, you have an embryo that's 50, 100, 1,000 cells, and only one cell is acquiring that mutation. And if you think about the frequency of KTS in the population, the rates at which mutations arise just by chance alone, if you were just sequence cells in culture, it seems more like it's a random chance event, rather than a mom or dad, well, it would be a mom, unknowingly was exposed to something that created a risk to her fetus at that time. That's, yeah, I can't say never, but I think that's exceedingly unlikely. I think more likely it is just accident of nature.
And that's good news for the moms. Thank you very much.
So running behind, maybe we'll have other questions you can chat with Dr.
I'm around, I'm looking forward to it. Thank you.